Good morning everyone. Myself Dr. Vishwa Raj Lal, Faculty of Biotechnology, Jamshedpur Women's College, Jamshedpur. Today we will discuss about protoplast fusion or somatic hybridization. Protoplast fusions or somatic hybridizations. Protoplast. The word protoplast was coined by Hanstein in 1880. Protoplasts are plant cells with the plasma membrane but without the cell wall. Cell wall completely removed using either mechanical or enzymatic process. So we can say that the entire plant cell without its cellulosic cell wall called protoplast. Next, protoplast fusions and somatic hybridizations. Technique in which in this protoplast fusion somatic hybridization, somatic fusions also called protoplast fusion is a type of genetic modifications in plants by which two distinct species of plants are fused together to form a new hybrid plant with the characteristics of both a somatic hybrid. Techniques in which two or more protoplasts are fused into a single cell is called protoplast fusion. In vitro fusion of plant protoplasts derived either from somatic cells of same plant or from two genetically different plants is called somatic hybridizations. Protoplast fusions and somatic hybridizations in plants are based on the following basic points. First, protoplast isolations. Second, fusion of protoplast. Next, cell wall regeneration by the fused product. Fusion of nuclei. Next, division of hybrid cells and their subsequent growth. Next, identification, selection or isolation by hybrid cells. Next, induction of organogenesis in the callus tissue, and lastly gives rise to mature plants. Then come to a protoplast fusions. There are two types, spontaneous and induced fusions. In spontaneous fusions there are mechanical fusions, and in induced fusion there are chemofusions and electrofusions. In spontaneous fusions, protoplasts fuse spontaneously during isolation process, mainly due to physical contact. Next, spontaneous fusion is strictly intraspecific and give rise to homokaryon. Then next come to a mechanical fusions. In this process, the isolated protoplasts are brought into intimate physical contact mechanically under microscope during micromanipulator and perfusion micropipette. This micropipette is partially blocked within 1 mm of the tip by the sealed glass rod, and in this way the protoplasts are retained and compressed by the flow of liquid. By this technique, occasional fusion of protoplast has been observed. Next, chemofusions. Fusion induced by some chemical agents such as sodium nitrate, polyethylene glycol, calcium ions, polyvinyl alcohol, etc. These chemical agents are called chemical fusogens. Chemical fusogens cause the isolated protoplast to adhere each other. It is non-specific. It is inexpensive. Next, electrofusions. Electrofusions. When we apply electric current to protoplast, this leads to pair chain arrangement of protoplast. The number of protoplasts within the pair chain depends upon the population density of the protoplast and the distance between the electrodes. When short DC pulses are applied, the membrane between the two protoplasts is break down and get the fused product. Fusion induced by electric current used an electric field of low strength. Electrofusion is easy to control. It is having fusion up to 100%. Next come to our uses. Used for combining the two complete genomes, exchange single or few traits between species. Studies of fusion product can give information about compatibility or incompatibility of the nuclei or cytoplasm. It is possible to transfer some useful genes such as disease resistance, nitrogen fixations, rapid growth, etc. So, protoplast fusions and somatic hybridations have opened up a new avenue in plant science. It is now a well-known fact that the somatic hybridizations in plants can be used in the improvements of plants. One of these is the production of hybrids, which is not possible through normal sexual fusions or fertilization process. In other words, it includes the formations of somatic hybrids between two species which are sexually incompatible. Thus, protoplast fusions provides a method of combining the different genomes of different genera and species with the potential of overcoming sexual incompatible barrier between plants.